So after having discussed the nerve, now let us discuss the muscle. Majority of this discussion will revolve around the skeletal muscle. Let's see what all topics we are going to cover. We will first discuss the NM junction, the neuromuscular junction or myoneural junction, then excitation contraction coupling, that will be followed by sarcomere, very important topic. Almost always the questions are coming from sarcomere as well as some questions from excitation contraction coupling. Then muscle contraction, the theories of muscle contraction and the factors influencing the strength of muscle contraction, where we will also discuss the tetanizing frequency of a muscle fatigue, motor unit and so on. And then energetics in the muscle, energy systems in the muscle, types of muscle fibers. I'm talking about the skeletal muscle fibers, red and pale. Finally, then we will discuss the smooth muscle and the cardiac muscle, the comparative features of skeletal versus cardiac versus smooth muscle. So that will be our course of discussion. Let's now start with the first topic. But before that, just as a basic discussion, muscle is of various types. Basically, the skeletal muscle, a part of musculoskeletal system, which is a voluntary muscle, and it's a striated muscle. Then we have cardiac muscle, which is also striated but involuntary. And then the smooth muscle, which is lining the viscera. It is non-striated, so appearance wise smooth, and it controls the visceral function. Right. We will start with the skeletal muscle, and the first topic is the neuromuscular junction. Now one might be wondering, what can be asked from this particular topic as an MCQ, a tough MCQ, because this is a very basic topic to understand. No, we very often we find that the topic is very basic, yet tough, tricky questions are asked. Let's see such questions and then we will have a discussion on it. Here is a neuromuscular junction, and if you are aware, there is acetylcholine which is in the nerve ending. This is a motor neuron starting from the spinal cord, and acetylcholine vesicles are there in its endings. So the question is, and look at the trick of the question, or rather the difficulty level. The question is related to the acetylcholine vesicle synthesis, formation. Select the correct statement of the following regarding the synthesis of the acetylcholine vesicle. A. Acetylcholine and the vesicle both are synthesized in the nerve cell body. Look, this is a motor neuron ending on the muscle. So this motor neuron is starting from the spinal cord, and it has a nerve cell body where synthesis of some of the material takes place. So so A statement is acetylcholine as well as the vesicle both are synthesized in the nerve cell body. B, acetylcholine as well as the vesicle both are synthesized in the nerve terminal itself, not in the cell body but in the nerve terminal. C, Acetylcholine is synthesized in the nerve cell body, but vesicle is manufactured locally. Or D, acetylcholine is synthesized locally in the nerve terminal and vesicle comes from the nerve cell body, nerve cell or nerve cell's body. So what do you think is the correct answer? Yes, it requires some understanding. Okay, and I'm sure most of you are thinking acetylcholine is synthesized in the nerve cell body and vesicle is manufactured locally in the nerve terminal. No, in fact it is exactly opposite. Look, let me answer this and then we'll discuss in detail, of course. Vesicle is a cell organelle, and just like any other cell organelle, the vesicle is also synthesized in the nerve cell body, and then it will be transported down this axon, down this nerve fiber, and then it will reach here. Whereas acetylcholine is very much synthesized locally. So let me just write down here. Vesicle is synthesized in the nerve cell body of this motor neuron, and acetylcholine is synthesized locally. If you remember, if you have read this topic previously, the enzymes for the synthesis and destruction of acetylcholine are very much present in the neuromuscular junction. You remember this, acetylcholine is released and after that there is cholinesterase enzyme which is going to degrade the acetylcholine. So it's very much located here, and therefore acetylcholine synthesis occurs locally in the neuromuscular junction or in the nerve terminal. Right. So that was just a sample MCQ as to how the MCQs can be formed. Let me ask you one more question and then we will start the discussion. Choose the correct statement of the following. This is related to the depolarization and repolarization and its relation with the muscle contraction and muscle relaxation. So choose the correct statement. Very often I've seen the students saying very casually, yeah, depolarization means contraction and yeah, repolarization means relaxation. No, that's a very, very wrong conceptual understanding. Depolarization and repolarization are they form the electrical excitation, and contraction is a mechanical event. Alright, therefore they are not the same. So with this, choose the correct statement. Okay, see, it's like this. The electrical impulse, the action potential, will travel through the nerve, it will enter the muscle and it will result in the muscle contraction. So in that context, this question has been asked. So choose the correct statement. A, depolarization leads to muscle contraction. B, repolarization leads to muscle relaxation. C, both D and repolarization together means action potential leads to muscle contraction, and D, both D and repolarization together means action potential lead to muscle contraction as well as relaxation. What's your answer? I'm sure you are thinking C, the action potential leads to muscle contraction. If not, then you will go to D. No, they are not the correct statements. The only correct statement out of all these is the statement A, that is depolarization leads to muscle contraction. All the others are incorrect statements. Why? We are going to discuss this in detail, partly in this segment and then later on again in detail in the excitation contraction coupling. That is where it will be required. So with that in mind, let's begin the neuromuscular junction. Here is a motor neuron. It starts from the spinal cord, it goes to the muscle, and here is a muscle membrane. So where the motor neuron is making a contact with the muscle membrane, this part is called as motor end plate. The part of the muscle membrane where the motor neuron is making a contact. Or it can be also called as postsynaptic membrane. Look, let's have a clarity on this. Synapse is a junctional region between a neuron and another cell. That another cell could be another neuron, or it could be a muscle, or it could be a gland. So neuromuscular junction is technically a synapse. Only thing is, for the sake of understanding, nerve junction, we will refer to that as synapse, and nerve muscle junction, although technically this is also a synapse, we will call it neuromuscular junction. And therefore we can very much call this neuronal membrane as presynaptic membrane, and the muscle membrane as the postsynaptic membrane, considering this to be a synapse. Right. What are the contents? In the motor nerve terminal, there are acetylcholine vesicles, and there are mitochondria. Look, again, mitochondria will generate the ATPs which are needed for the synthesis of acetylcholine. So acetylcholine is very much synthesized locally over here. There are two more contents here, or parts we can say. One is voltage gated calcium channels. Voltage gated calcium channels. Why do we need these voltage gated calcium channels? It's because the vesicle, its membrane is negatively charged, and the presynaptic membrane, just like any cell membrane, is also negatively charged. And what are we expecting? When the impulse arrives at the nerve terminal, we expect this vesicle to fuse with the presynaptic membrane and then release the acetylcholine. But how can that fusion occur? Because both are negatively charged, it will be repulsion. And therefore, we need that there is a bridge formation between a vesicular membrane and the nerve membrane, presynaptic membrane. That bridge formation is carried out by the calcium. But as you are aware, calcium cannot be kept high inside the cell, in the cytoplasm. And so, it so happens that when the impulse arrives, the voltage gated calcium channels will sense it. They will open. The calcium from the ECF will enter the terminal, and then the vesicle will migrate towards the membrane and fuse with the membrane thanks to this calcium which has come in through the voltage gated calcium channels. And the other thing that is present here is the release sites where the vesicle fusion will take place. Coming to the muscle membrane, what all things are present here? Okay, there is acetylcholine receptor. So acetylcholine receptor, it is a ligand gated ion channel. Let's write it here. We are talking about the acetylcholine receptor. So first point, it's a ligand gated channel. I'm trying to say that this protein has got two parts. One part behaves like a receptor for acetylcholine, and when acetylcholine combines with the receptor portion, then the associated channel will open. The gates are flung open and the channel will open. So ligand gated ion channel. If you remember, we have discussed this in previous section that ligand gated ion channels are pentameric proteins. They are pentamers. The other thing that you should know is that this particular protein or this particular channel is a non-specific cation channel. Non-specific cation channel. It should be emphasized because we have seen sodium channels, potassium channels, calcium channels. They were very specific. They allow the movement of only that specific ion. But here is a channel which is a non-specific cation channel. Means what? When the acetylcholine is released and it combines with the receptor portion and the gates are opened, the same channel will allow the sodium movement, that is sodium influx, and same channel will allow the potassium movement, that is potassium exit. You know, potassium exits by concentration gradient. So that's a non-specific cation channel. This was about the postsynaptic membrane. Let's now see the events that occur at the neuromuscular junction. Impulse arrives at the neuromuscular junction. Well, impulse means depolarization. So please remember, if you're thinking action potential, it includes depolarization and repolarization both. So from now on, you must keep this in mind that when we say electrical excitation or a traveling impulse, we are essentially talking about the depolarization. It is the depolarization which travels through the nerve. It is the depolarization which is a traveling impulse. It travels through the nerve. It enters the muscle. It's going to cause release of calcium into the muscle's sarcoplasm, and that will initiate the contraction. So depolarization leads to muscle contraction is the correct statement. Then how do you look at the repolarization? Look, depolarization, I am talking about the depolarization, traveling impulse. It is taken as a traveling impulse. Repolarization is taken as recovery from excitation. It should not be seen as a part of traveling impulse. Repolarization will occur point by point by point. The recovery of the membrane from its excitation, it occurs at every point by point. It should be seen like that. Okay. This is a concept that you must understand. Only when you understand this, you will understand what happens in the case of cardiac muscle, how the depolarization and repolarization spread or occur. But remember, repolarization is recovery from excitation. It does not travel. It only occurs point by point. Whereas depolarization is to be taken as the traveling impulse. It travels through the nerve, enters the muscle, causes contraction. So if you were thinking repolarization causes relaxation of the muscle, no, that's not correct. Relaxation of the muscle, as we will see later again, is when the one more depolarization does not come into the muscle, then first depolarization has caused muscle to contract, and then if one more excitation, depolarization does not come, then the calcium that was released into the sarcoplasm, it is pumped out of the sarcoplasm into the endoplasmic reticulum, and that will lead to fall of calcium and relaxation of the muscle. So relaxation is not linked to repolarization or something. All right, let's see the events. Impulse arrives at the motor nerve terminal. It causes release of acetylcholine into the junction. Okay, each impulse, this is just a factual information, each impulse causes release of about 60 vesicles of acetylcholine. and each vesicle contains almost 10,000 molecules of acetylcholine. So each vesicle containing 10,000 molecules of acetylcholine, and such 60 vesicles almost are released with every impulse. Such a high release of acetylcholine at the neuromuscular junction. So when such a high release occurs and acetylcholine then combines with its receptor portion of this protein, it combines with the receptor, it traverses the gap. And of course, one more point, acetylcholine is released by exocytosis. That's very obvious here. Vesicle will release the contents. When it combines with the receptor, what happens is the channel opens. The associated channel will open. And as we have mentioned, there will be sodium influx and potassium exit. Both will happen through the same channel. But sodium influx predominates even if we are allowing both ions to go through the channel. Sodium influx will be a large event and potassium exit will be relatively small event. Sodium influx predominates, and that will result in the net entry of positive charges. I mean 100, I'm just giving the example, 100 sodium ions coming in and only 5 potassium going out. So net entry of 95 positive charges. Just an example. The question is why? Why it happens like that? Again, a concept, and we have actually discussed this concept in the previous section. Look, the muscle was resting. This muscle, skeletal muscle, has an RMP of minus 90 millivolts. It was resting at minus 90 millivolts. Now the acetylcholine has caused the opening of these channels and sodium and potassium have moved. If you remember, the equilibrium potential for sodium in the case of muscle is plus 61 millivolts and equilibrium potential for potassium is minus 96 millivolts. And if you remember, we have made a very important statement. If the membrane potential is closer to the equilibrium potential of an ion, then the ion movement will be less. Ion will be either in equilibrium or its movement will be very, very less. And if the membrane potential is far away from the equilibrium potential for an ion, then the ion movement will be large. Please keep this in mind. And therefore, at minus 90 millivolts, if you allow both sodium and potassium to move, potassium movement will be not that marked, because see, the membrane potential is closer to the equilibrium potential for potassium, but it is far away from the sodium's equilibrium potential, and therefore the pull on the sodium will be much larger to move in compared to the pull on potassium to go out, which will be very less. And that's the reason why sodium movement or sodium entry predominates over the potassium exit, potassium going out, and therefore there would be net entry of positive charges, and that will result in depolarization. This depolarization will travel into the muscle and it will result in muscle contraction. These are the events that occur in the neuromuscular junction. Now let me add one or two more features related to this. I said that depolarization occurs. Does it occur instantly, immediately? No. There is first development of a small localized potential, and when it reaches threshold, then it will develop into an action potential, means complete depolarization will take place. So please remember, action potential, wherever it is happening, whether it's a sensory neuron in the skin or whether it is the neuromuscular junction, it does not occur suddenly. First, there is a precursor potential, and when that precursor potential is large enough, it will reach the threshold, the membrane will reach threshold and will develop the full action potential. So that precursor potential in this case is called as end plate potential. So I always maintain this particular example is my favorite example. Imagine the membrane bursting into action potential, but it does not happen suddenly. First it reaches threshold slowly and then it bursts into action potential. So somewhat similar to the firecracker. I like to give this example of firecracker. Firecracker just does not burst instantly. There is a slow burning first, and then when it reaches a threshold, then it bursts instantaneously. Same thing happens here, same thing happens anywhere wherever there is action potential being generated. That first a localized potential will develop, and when the membrane reaches threshold, then it develops into action potential. So end plate potential is a localized potential. It is considered to be a forerunner of the action potential, means first this will develop, membrane will reach threshold, and then action potential. So let's see some of the few things about the end plate potential. Its amplitude is about 40 millivolts. Why it is necessary? Imagine that muscle threshold to develop action potential is minus 55. So minus 90, we'll just show it graphically. Minus 90, muscle is resting, gradually reaching threshold, which is minus 55, and then developing the full action potential. Minus 55 means there has to be a depolarization of about 35 millivolts. You can see here, positive charges that are coming in should take the membrane at least up to minus 55, means the threshold should be reached. Whereas what we are saying here, end plate potential, whenever it develops, it's about 40 millivolts. It means what? It means definitely threshold will be reached. It's a 40 millivolts of positivity which is coming in, or means positive potential, which is or positive ions which are taking the membrane towards less negativity or more positivity. So definitely membrane will reach threshold and definitely action potential will be generated. Why am I emphasizing this? It's because, now the next statement is important, so please listen to it. It's because whenever the impulse is reaching the neuromuscular junction, the meaning is that the cortex, our cerebral motor cortex, has made a decision that this muscle needs to contract. Definitely it should contract, and therefore such a large precursor potential is developed, so that muscle will definitely reach the threshold, will cross the threshold, and will definitely generate one action potential and will form one muscle contraction. So cortex has made the decision to contract the muscle, and therefore, now what is the big deal in this that I am telling you? The big deal is there are similar precursor potentials in the CNS synapses. Just as this is the synapse, there are synapses in the CNS, and in the synapses in the CNS, there is a similar precursor potential which you may have heard of. It's called EPSP, excitatory postsynaptic potential. It develops on the postsynaptic membrane, just like the muscle membrane over here. In the CNS synapses, the postsynaptic membrane. So EPSP. You know what is the amplitude of EPSP? Just about 2 millivolts, 4 millivolts, 5 millivolts. That's it. It means a single EPSP will not convert into a single action potential. Point is important. One impulse in the motor neuron, which we are discussing right now, will convert or will form one large EPP on the muscle membrane, the end plate potential, 40 millivolts, and it will definitely convert into one action potential in the muscle and will result in one muscle contraction. Whereas if you compare the similar potential in the CNS synapses, or in the case of receptor potential in the skin, localized potentials, similar way, but 2 or 4 or 5 millivolts. That means a single action potential in the presynaptic neuron will give rise to one EPSP, but that will not convert into one action potential because it is small, 2-4 millivolts. The postsynaptic neuron will not reach the threshold and will not generate the action potential in the postsynaptic neuron, which means multiple EPSPs have to summate. There has to be a summation of EPSPs in the CNS we are talking, so that they get added, like 5 millivolts will make a 15 millivolts, and then the postsynaptic neuron will reach the threshold and will convert into one action potential. So compare EPP, one EPP converts into one action potential because we need a definitive contraction of the muscle, whereas CNS always goes on learning by trial and error, and therefore it's not given in a very definitive manner. So multiple action potentials in the presynaptic neuron, multiple EPSPs will be generated, they will get added up to form one action potential in the postsynaptic neuron in the case of CNS synapses. Therefore, that comparison will give you a fair idea of this concept of EPP. So remember some of the properties of EPP. It's a localized potential. If you compare this with the action potential, let's compare this. EPP is localized. Action potential is a propagated potential. Meaning what? Look, the channel opened here and positive charges have come in. So those positive charges will form a local potential over here in a small area of the muscle membrane. It's a localized, but when it converts into an action potential, then action potential travels in the muscle. It's a propagated potential. The action potential is a propagated potential. Once it develops at one point, then it occurs in a self-propagated manner, self-regenerative manner. Whereas EPP, wherever it develops locally, as the ions will travel passively, the strength of EPP will decrease. It's just passive spread of charges. That's it. EPP is considered to be a monophasic potential, whereas action potential is a biphasic potential. End plate potential is monophasic in the sense that it's a single phase of depolarization from -90 to -45 or 50, the 40 millivolt depolarization, that's it. Whereas action potential contains electrically two opposite types of events. Depolarization means inside becomes positive, repolarization means inside again becomes negative. So electrically two opposite types of events. And these potentials, EPP or EPSP or receptor potential in the skin, these precursors are said to be the graded potentials. Means what? They show gradations. I mean, just giving you an example. If one vesicle is released of acetylcholine, 2 millivolt EPP generated, or rather 2 millivolts of EPSP generated. Two vesicles released, and let me again say, this is an example, okay? Two vesicles released, 4 millivolt EPSP generated. Three vesicles released, 6 millivolts of EPP generated. So this is just an example. I've already mentioned to you, lots of vesicles are released every impulse. But the point that I was making was gradations are seen in the EPSP or receptor potential, but action potential is all or none, no gradations. It's all or none phenomenon. I mean, if it develops with full amplitude, all. And if threshold was not reached, then nothing. So all or nothing principle by action potential, and the opposite of that is graded potential. So please remember, EPP, EPSP, receptor potential, these are all graded potential. They show gradations. It depends on the strength of the stimulus and how much positive charge is going to come in. Right. There is one more additional factor, MEPP. Miniature end plate potential. MEPP, miniature form of the end plate potential. Look, when the impulse arrives at the neuromuscular junction, there is development of EPP. But when impulse is not arriving at the neuromuscular junction, muscle is at rest, it's not supposed to contract, and still, as an after effect of those impulses which have already occurred, there will be asynchronous release of acetylcholine at the neuromuscular junction. Asynchronous release of acetylcholine, and it is in minute quantities. Therefore, that asynchronous release of acetylcholine in minute quantities and opening of channels in the muscle membrane will result in a very small form of EPP. Therefore, it's called as miniature end plate potential. It's in microvolts. EPP was in millivolts. Most of our potentials that we discuss are in millivolts. MEPP is in microvolts. What is the significance? Generally, there is no significance attached to it because it's not going to cause muscle to contract. So just remember, there is some release of acetylcholine even when impulse is not arriving at the nerve terminal, and it develops the potential on the muscle membrane called as MEPP. Right. These are some of the aspects related to the neuromuscular junction or myoneural junction. Let's see the applied physiology. Autoimmune diseases related to the neuromuscular junction. There are two autoimmune diseases which we should be aware of. One is, of course, myasthenia gravis. Myo is muscle, asthenia is weakness, gravis is grave. So grave weakness of the muscle. The other one is Lambert-Eaton disease. Lambert-Eaton myasthenic syndrome also it can be called, LEMS. Lambert-Eaton myasthenic syndrome. What is common? Common to both is the fact that they are both autoimmune diseases and they both result in muscle weakness, profound weakness of the muscle. So, what's different? In Lambert-Eaton disease, those antibodies are developed against the calcium channels in the presynaptic terminal. So essentially this is a presynaptic disease. Whereas myasthenia gravis, the antibodies are developed against the nicotinic acetylcholine receptor. Well, we probably did not mention this at the start of this section. The acetylcholine receptor, which is of two types, you are aware, nicotinic and muscarinic. This receptor is a nicotinic receptor. Remember, nicotinic receptor is present. It's present in the neuromuscular junctions. It is present in the ganglia for the ganglionic transmission, and it's present in the CNS neurons. So this is a nicotinic receptor. Antibodies develop against nicotinic acetylcholine receptor in the neuromuscular junction. All right. What are the features of this myasthenia gravis? There is weakness, tremendous weakness, because if those receptors are either occupied by those antibodies or destroyed, so suppose a muscle has 100 muscle fibers, and in 90 muscle fibers the receptors have been destroyed. We are left with only 10 fibers to contract, and that means 10 fibers will produce a very weak contraction of the muscle, and also there will be easy fatigability. Muscle will fatigue early, naturally, because 10 fibers are made to perform the job of 100 fibers of that muscle. The muscle is made up of 100 fibers, but only 10 are contracting. Only 10 fibers have receptors still intact. So obviously the muscle will fatigue early. These are the symptoms. Similar symptoms are also seen in the Lambert-Eaton disease. Only difference is with continuous contraction of the muscle. Now I am making a comparison of the two. Okay. The differentiating feature between the two. Lambert-Eaton disease, antibodies against calcium channels. So calcium influx will be defective, and therefore release of acetylcholine also will be defective, and therefore only a few fibers where it is still left, those calcium channels, those fibers will contract. But the point is, with repeated muscle contraction, with repeated continuous muscle activity, the myasthenia gravis symptom will become worse. It worsens with repeated muscle contraction, because those fibers which are contracting, even they will eventually go into fatigue. Acetylcholine, whatever it was, it will be exhausted, and then the symptoms will become worse and worse with repeated continuous voluntary motor activity. Whereas in the case of Lambert-Eaton, it has been observed that with repeated continuous muscle contraction, a point comes where suddenly the contraction becomes better, the contraction becomes stronger. Okay, see, in both cases it was weak contraction only to start with, but in Lambert-Eaton disease it is observed that over certain duration, if it's a continuous muscle contraction going on for a few minutes, let's say, there is improvement in the strength of muscle contraction. Why this happens? It happens because if there was a continuous signaling to the muscle, and wherever the calcium channels were still available, the calcium will go on entering, and calcium will at one point enough calcium will be accumulated in the presynaptic terminals. That then will cause a better contraction. So starting with weak, but at one point an improvement in the contraction. That's seen in the Lambert-Eaton. Whereas myasthenia gravis, it goes on worsening and worsening. Right. That was the applied physiology related to the neuromuscular junction, and of course you will read it in pharmacology, what drugs are given for this. So that will be a part of pharmacology discussion. But this is the neuromuscular junction and the transmission of impulse. The next topic will be excitation contraction coupling. That's excitation contraction coupling, commonly known as EC coupling. Another favorite topic with the entrance exams. Lots of questions have come in the recent past. For example, DHPR, this dihydropyridine receptor. Select the true statement about DHPR. And the options are, it is T type of calcium channel in the T tubule. It is L type of calcium channel in the T tubule, or T type of calcium in the L tubule, or L type of calcium in the L tubule. So basically, let me just read it out again for you. There are two types of tubules, as you are aware, in the system of sarcoplasmic reticulum: T tubule and L tubule. And incidentally, the calcium channels also are of various types: T type of calcium channel and L type of calcium channel. And that is the entire confusion. What type of calcium channel is it? The DHPR. And it's present where? In the T tubule or L tubule. That's one question which has been repeatedly asked in the FMGE exams and other entrance exams. Another question which has come once or twice is that a 12-year boy was to be operated upon, and just before the operation, just before the surgery, succinylcholine was administered, and suddenly the boy started shivering. So what is this condition, or where is the defect basically? And the options given were DHPR, RYR, dystrophin, titin. So RYR, genetic defect resulting in malignant hyperthermia. That was the question based on. And of course, DHPR is present in the T tubules, L type calcium channel in the T tubules. Let's now discuss all this in detail. Now, first of all, what is the concept of excitation contraction coupling, EC coupling? Let me just first tell you the concept and then we will describe it. Look, we have discussed neuromuscular junction. The impulse travels through the nerve, the motor neuron, and it crosses the neuromuscular junction, and then it excites the muscle membrane. From this point onward, we got to discuss the excitation contraction coupling. That is, the electrical excitation has traveled through the nerve and it has entered the muscle now. How will it result in the process of contraction? So, electrical excitation. Excitation is an electrical type of event, and it's going to result in the contraction, which is the mechanical type of event. Actin-myosin interaction is the mechanical type of event. So, electrical and mechanical event is coupled. It's clubbed together, or it's coupled. Somewhere the electrical excitation ends and the mechanical process of contraction starts. And these two have been coupled. Two different types of events have been coupled, electrical and mechanical, clubbed together. That is the concept of excitation contraction coupling. First point is that calcium is the coupling agent, because you know, where the electrical excitation ends, there is a release of calcium, and this calcium then initiates the mechanical process of contraction. So calcium is the coupling agent. It couples these two events. Second point is that the coupling occurs in the sarcotubular system of the muscle. there is a system of tubules in the muscle. Sarco, whenever we now use the term sarco, it will mean related to the skeletal muscle. For instance, sarcoplasm will be the cytoplasm of the muscle fiber. Sarcolemma will be the muscle fiber membrane. And sarcotubular system is the system of tubules in the muscle. It consists of two types of tubules. Let's draw it here. Here is a muscle, and the two types of tubules. The T tubule, that is transverse tubule, and the L tubule, that's the longitudinal tubule. These two are the types of tubules in the muscle. So muscle length is like this, and therefore L tubule is the longitudinal one along the length, and this will be the T, the transverse tubule. Therefore, first point that we said is that the coupling will happen in this sarcotubular system. Second point is that you can see here there is one end of the T tubule and two blind ends of the L tubules on either side. This is called as triad because it's three, so triad. The coupling occurs in the triad. Another point is that these expanded ends of the L tubules, which are on the either side of the T tubule, these are called as cisterns or cisternae. And these are the storehouse of intracellular calcium. So here is the calcium stored in the cistern. This calcium will be released into the sarcoplasm, which is here, and this will initiate the process of contraction. Well, by the way, behind this, into the muscle fiber, there are those thin and thick filaments, and their sliding or their interaction will result in muscle contraction. Another point, just note it here, then we will discuss in detail. In the case of skeletal muscle, there are 2 T tubules per sarcomere. I have deliberately shown 2 T tubules per sarcomere. So that's a skeletal muscle. These T tubules are in front of the junctions of I bands with A bands. When we will discuss sarcomere, we will describe these bands, and then again we can recall this. But for now, just remember there are 2 T tubules per sarcomere in the case of a skeletal muscle, and they are present in front of the junctions of I bands with A bands. In the case of cardiac muscle, there is only 1 T tubule per sarcomere, and that 1 T tubule in each sarcomere is lined in front of the Z lines. That's the difference between the skeletal and cardiac muscle. One more fact that you can remember here is T tubules are deepest in the case of skeletal muscle. They are little shallow in the cardiac muscle, and in the smooth muscle they are almost rudimentary. T tubules are rudimentary. That another difference you can remember. Now the most important part of the discussion, the 2 muscle proteins which are involved in the coupling. In the membrane of the T tubule, there is a protein called as DHPR, dihydropyridine receptor, because it binds with a category of drugs called as dihydropyridines. They are calcium channel blockers, CCBs. So that's dihydropyridine receptor. And in the membrane of the L tubule, there is another protein called as RYR. That is ryanodine receptor. These proteins are involved in the process of coupling. Ryanodine receptor is going to act like a calcium release channel. So it's something like this, that the nerve sends the impulse. The impulse will cross the neuromuscular junction. Here is the neuromuscular junction. It will be crossed. Muscle membrane will excite, and then the depolarization will travel deep into the muscle via T-tubule. DHPR has a voltage sensor. It has a voltage sensing mechanism. We can say it is a voltage sensitive calcium channel. It's a voltage sensor or voltage sensitive calcium channel or L type of calcium channel. Okay, now when the depolarization travels via the T-tubule, it will be sensed by the DHPR, because the DHPR has a voltage sensing mechanism and depolarization is a voltage change. Then it will interact with the RYR. It's a mechanical interaction. In the case of skeletal muscle, this is simply a mechanical interaction, that the RYR will be opened up by the interaction with DHPR. So depolarization came in, it was sensed by DHPR, DHPR interacts with RYR, RYR opens and calcium will be released. Calcium which was stored here will be released into the sarcoplasm. This calcium will combine with the troponin C, and that will initiate the process of contraction. So up to the release of calcium, the events were electrical, depolarization, electrical event, and after the release of calcium, the events are mechanical interaction between thin and thick filaments, and therefore calcium is the coupling agent. So here is the calcium released into the sarcoplasm. Calcium will combine with troponin C. You know, as I said, behind these tubules, there are those thin and thick filaments in the sarcomere. And on the thin filament, there is this troponin C. So calcium will combine with that, and that will initiate the actin and myosin interaction. Muscle contraction begins. That is the process of excitation, contraction, coupling. Let's just draw a flow chart and summarize this again. Depolarization travels deep into the muscle via T-tubule. Then it will be sensed by the DHPR. DHPR then interacts with RYR, ryanodine receptor, which is a calcium release channel. So calcium is released into the sarcoplasm, and then this calcium combines with troponin C. That will cause actin myosin interaction. Muscle contraction begins. Right. What is the cause for muscle relaxation? Muscle relaxation will happen when calcium level in the sarcoplasm decreases, falls. How does that happen? Look, one depolarization has resulted in one muscle contraction. If one more impulse does not come in, if one more depolarization does not come, then this calcium is pumped back. It is pumped back in those cisternae by a pump, an ATPase called as SERCA. Sarcoplasmic endoplasmic reticulum calcium ATPase. It's an active transporter, ATPase. It pumps the calcium out because there was no next impulse coming. And since the calcium is pumped out of the sarcoplasm, its level falls in the sarcoplasm, and that causes cessation of or stoppage of actin-myosin interaction. Actin-myosin interaction will stop and the muscle will relax. So that is the mechanism of muscle relaxation. So remember, I had asked this MCQ in the previous section. True statement. Depolarization leads to contraction, repolarization leads to relaxation. Let me just reiterate it once again, because it's a concept, you are going to need it forever. The only correct statement is depolarization resulted in muscle contraction. Depolarization was sensed by the DHPR and then it resulted in muscle contraction. Relaxation, I know most of you are still thinking, oh, repolarization means relaxation. No, please clear out that concept. Relaxation is because of the calcium pumped out of the sarcoplasm, and sarcoplasmic calcium levels decrease, and that results in muscle relaxation. That brings about the muscle relaxation. Then repolarization. How do you look at the repolarization? We have described this also, that repolarization is recovery from membrane excitation. I mean, depolarization has traveled through the nerve, then every point will recover from that excitation, and that's called as repolarization or recovery from excitation. Alright, that was the excitation contraction coupling and all the events associated. Let's see the applied aspect of this. First point, malignant hyperthermia. Malignant hyperthermia. Look, we have used the term malignant as in malignant hypertension, malignant hyperthermia, but it does not necessarily reflect some cancerous condition. Over here, malignant hyperthermia is a genetic disorder with the defect in the gene that codes for RYR. So it's called as RYR gene, and therefore RYR, the ryanodine receptor, is defective. Normally, it would not cause any problem under routine conditions. But what has been observed is, when these people are administered inhalational anesthetics like halothane, ether, or if they are administered muscle relaxants like succinylcholine, then, and that is before surgery, just before the surgery, this person was administered anesthetic gases or skeletal muscle relaxant like succinylcholine, then what happens is there is excessive release of calcium through the RYR because it is defective. And because of the excessive release of calcium, there is massive muscle contractions happening. Muscle contractions happen in a very oscillatory rhythmic fashion. So it appears like a shivering, rhythmic muscle contractions. Just before the surgery, the patient starts rigorous muscle contractions. And because of those contractions, there is increased muscle metabolism. There is increased heat production. Therefore, this term is called as hyperthermia. Increased temperature, increased heat production. That's malignant hyperthermia. It will exhibit tachycardia. Of course, if the heat production, increased metabolism, there will be tachycardia, increased body temperature, shivering appearance. So that all will be manifested, and it's treated with the uncoupling agent. Dantrolene sodium is considered to be the uncoupler. It uncouples the electrical excitation and mechanical contraction. So this drug acts on the EC coupling. Well, additionally, you may also remember two more diseases which are related to the EC coupling. Central core disease. Central core disease is also a result of genetic defect of RYR. Then Brody's disease, again a disease related to the EC coupling. And what is common in malignant hyperthermia and central core disease and Brody's disease is excessive release of calcium because of the defective RYR. And then the manifestations will vary. For example, central core disease means that excess calcium release into the sarcoplasm might be taken up by the mitochondria. They will be studded with the calcium stones. Eventually they will get degraded, leaving behind the core in the cell, in the cytoplasm. So that's central core disease. Anyways, the point was, remember the names at least of these diseases. If you see in the options in the MCQs, you might recall that, okay, this is related to the excitation contraction coupling. Now, having discussed all this in the skeletal muscle, now is the time to also discuss the EC coupling in the cardiac muscle and see the differences between skeletal and cardiac muscle. So first point, let me just write it here, EC coupling in the case of cardiac muscle. Let's draw the sarcotubular system in the cardiac muscle. The T-tubule in the case of cardiac muscle is shallow but wide. Shallow but wide. Remember, in the skeletal muscle, it was little narrow and deep. But because of this kind of a structure, this is a T-tubule, its surface area is much more as compared to the skeletal muscle T-tubule. And the second point is, this T-tubule is an invagination of the muscle membrane. We are talking about the cardiac muscle. It's an invagination of the muscle membrane and it is a storehouse of ECF calcium. So please remember, ECF calcium is stored in the T-tubule. Second, the L-tubule. So just like in the skeletal muscle, there is the L-tubule in the cardiac muscle. And yes, note this point, I have drawn only one T-tubule here. As we mentioned, one T-tubule per sarcomere in the case of cardiac muscle, two T-tubules per sarcomere in the case of skeletal muscle. And where is it located? In front of the Z lines, which we will discuss in sarcomere. Right, so this is RYR, similar to the skeletal muscle. Let's see what happens in the cardiac muscle. Impulse arrives. This is a impulse arrives in the cardiac muscle. Of course, it's a conducting tissue which carries the impulses into the cardiac muscle. And there are nerves which control, the vagus nerve and the sympathetic nerves. When the impulse comes in the muscle and it travels through the T-tubule, now the important difference, very important difference coming up. Here is a DHPR. And the DHPR, as we have said, it is L-type voltage sensitive calcium channel. Alright. So, when the impulse comes, which is a voltage change, it will be sensed by the DHPR and DHPR itself opens and the ECF calcium enters through the DHPR because it's a calcium channel. Extracellular calcium enters and it causes further release of calcium through the RYR. Okay, this is called as calcium induced calcium release. Calcium induced calcium release. Look, in the case of skeletal muscle, it was depolarization induced calcium release. The depolarization went into the muscle and it was sensed by DHPR and DHPR mechanically interacted with RYR and entire calcium came from the intracellular store. But in the case of cardiac muscle, when the impulse comes in, it is sensed by the DHPR and DHPR itself opens. Let's just write it down. When impulse arrives in the T-tubule, DHPR itself opens after sensing that impulse. Then ECF calcium enters through the DHPR and it causes further release of calcium through the RYR. So this has been called as calcium induced calcium release, and in the case of skeletal muscle it's called as depolarization induced calcium release. And then rest of the things are similar. This calcium will then initiate the contraction of the cardiac muscle. Right. So what are the differences that we saw? Just summarize again. 1 T-tubule per sarcomere. DHPR is actually a channel here, just as we have said in the skeletal muscle, but there it does not open to cause calcium entry from outside. Here it actually opens and the ECF calcium goes in and causes further release of calcium. So calcium induced calcium release. What is the applied aspect of this? What is the application of this knowledge? One is, you noted this point that ECF calcium is involved in the process of cardiac muscle contraction. It was not involved in the skeletal muscle contraction. So changes in the ECF calcium levels, they will influence the cardiac contractility, but they will have no influence on the skeletal muscle contractility. Yeah, I just repeat once again. For the skeletal muscle contraction, all the calcium came from the intracellular stores, those cisterns, L tubules. Whereas in the case of cardiac muscle, ECF calcium entered and that ECF calcium is causing further calcium release from the intracellular stores. So ECF calcium is jolly well involved in the process of contraction, and therefore changes in ECF calcium, they do influence the cardiac contractility, but they won't have any influence over the skeletal muscle contractility, as all its calcium came from the intracellular store in the case of skeletal muscle. And the second difference, second applied physiology can be the calcium channel blockers. You know, there is a particular variety of calcium channel blockers, the dihydropyridines, which I mentioned some time back. The prototype is nifedipine. Okay, let me just add one point here. Changes in the ECF calcium, they do influence the cardiac contractility as well as smooth muscle contractility. Okay, smooth muscle mechanism is also similar to this, which we discussed just now. So please remember, extracellular calcium will influence the cardiac and smooth muscle contraction, but not the skeletal muscle contraction. And the second, the prototype nifedipine, the dihydropyridines which combine with this receptor, they will have influence on the cardiac contractility and as well as smooth muscle also is influenced by them. The DHPRs can be acted upon by these drugs, so they can cause vasodilation as well as decrease in the cardiac contractility. They are used as antihypertensives, but they will not influence. Of course, please make a note here. I know that you might read in pharmacology that there are cardioselective calcium channel blockers and non-selective calcium channel blockers. So forget about that for now. I am just talking about these drugs which can influence the cardiac contractility. In this category, there are certain cardioselective drugs. So they will influence the cardiac contractility but they will not influence the skeletal muscle contractility, because the structure of DHPR is different in cardiac muscle and different in the skeletal muscle. So these drugs, they do act on the cardiac DHPRs, but not on the skeletal muscle DHPR, not the skeletal muscle DHPR. They can act on the cardiac muscle, they can act on the smooth muscle to cause vasodilation, they can act on the cardiac muscle to decrease the contractility, but they cannot act on the skeletal muscle. So that was the applied aspect of the excitation contraction coupling.